Sorry, I just want to make sure it's recorded. Okay. Now no one knows who we are, which is great for the recording. Yeah, and then interrupt at any time. There's an interactive session at the end. We have a small amount of data and some code you can run. If you don't want to do that, you don't have to, but we thought we'd provide it. And then the sort of like output at the end of this, also I'll make all the code that I use available to run the things that I've done on GitHub for you to use. It's all based in Terra with the workflow description languages or WDLs. If you don't use that, you can at least look at the R script and the code that sort of underlies it if you run it locally or on Acre. And then Marina kindly made like a little tutorial for us, so it was great. So basically we thought about like, let's say off the sequencer, you get your methylation data. Vantage will provide you with a CX report file. Basically, what do you need to do to go from CX report file to biologic insights? Essentially this process, which is kind of convoluted, but we'll walk you through each step of them. So we split it up to talk about it in each section. So, you need to quality control your data to basically make a matrix that represents the methylation data. Once you have that, you obviously will have your phenotypic and covariate files to perform the EWAS. But I think the thing that I'd never understood before I started doing methylation is that the cell proportions in your blood actually matter a lot when you're testing to see what happened, like what's associated with your trait. And if your outcome changes cell proportions, then your EWAS will be completely confounded by cell proportion differences. So that like leads to this sort of like blue area that we dived into. And then then we'll split the insight section to various ways we can use methylation data and it's the summary statistics downstream. And then we have an expert in epigenetic aging. You said you're a geroscientist. So I think clocks and risk scores are very much a way to, take an EWAS that someone else has done or EWAS you've done previously and then apply it to new data that you're generating to make predictions about, an individual person from their CX report file. Yep. Cool. Diving in. Yeah. Yep. So yeah, we'll start with background and quality control.
Okay, perfect. Okay, so just a quick primer on DNA methylation because I'm not sure kind of where everyone is at and understanding this. So DNA methylation, these are epigenetic modifications that bind to the DNA. They don't change the underlying sequence of the DNA, but they do change how the DNA is read. And so here we're talking specifically about 5-MC methylation. You don't really need to know the specifics, but if you're really deep in the methylation world, you will hear about two types of methylation. This is just where those methyl groups are binding to cytosine. And so typically when we're talking, I'm just going to use the shorthand and say DNA methylation, that means 5-MC. And so this is transcriptionally inhibitory typically. So if we have methylation, you can think downregulation in gene expression. There's another type of methylation that people have been excited about recently, but it occurs far less frequently, so we're not going to get into it. And also the twist panel doesn't capture it. But if you're really excited about different types of methylation, you can chat with us about these. And then when we're talking about DNA methylation today, we're going to be talking about CpG methylation. So this is methylation that happens in a CG context. There are other types of methylation that happen with between a cytosine and other base pairs. That's also less frequent, and it's less frequent in mammalian genomes in particular, and also really widely not as applicable in adult phenotypes. So if you're thinking about development, then it might be applicable, but we're going to focus on CpG methylation, so our shorthand will just be DNA methylation. Okay, and DNA methylation happens in the context of the broader chromatin and can interact with chromatin accessibility or chromatin marks to alter how the genome is packaged and how the DNA is read. So methylation can mechanistically impact gene regulation. There's been some really elegant studies in the past and a couple recently inducing methylation in specific areas of the genome and then observing that gene expression is mechanistically up or down regulated, increased or decreased. Methylation is also of interest because it is a very stable mark typically. So typically methylation can persist for many, many years. If we're thinking, I think Yash and I both study humans now, so we're going to be primarily assuming that everyone's studying humans. But yeah, methylation can be stable for long periods of time, but it can be malleable, particularly in certain areas of the genome, and that's of interest. So it is really expected to be a mechanism by which environmental perturbations can influence the genome and gene expression and then downstream phenotypes, particularly in immune cells, but really across tissue. So it's environmentally modifiable. Smoking has a big impact on methylation, famine, et cetera. So there's early developmental applications, and then also obviously it's implicated in aging. It's a DNA methylation or hypomethylation globally is a hallmark of aging, so also of interest to kind of both ends of the lifespan here. And then of course DNA methylation and other epigenetic modifications are the way that every single cell in our body can have the same or approximately the same genetic code, but have wildly different functions. So just on the bottom here is kind of a little schematic showing that while most methylation or CpG sites cluster together in the genome, there are some areas that are going to be more dispersed. So typically when methylation kind of co-occurs very locally in CpG islands, you have more sites and that will have a larger impact. So there's all sorts of fun things to think about, but often if we're thinking about mechanistic gene regulatory methylation, that's often thinking about methylation in promoters and enhancers. But if you're in the aging world, you're probably thinking a lot about methylation in other areas of the genome as well. Okay, so in the kind of recent history of methylation, there's the common types of methylation sequencing, DNA methylation sequencing that have been used. Oh, you're good.
Just don't want
Yeah, it was like
floating.
right.
Maybe that's
Perfect. Have been either microarrays. So people in this room might be really familiar with like the 450K microarray or the EPIC array. There's a version 1, a version 2. And then a lot of non-human studies, so I used to work in primates, employ reduced representation by sulfite sequencing. And so that essentially chops up the genome with an MSP1 digest and then measures methylation. And both of these use sodium bisulfite sequencing to convert unmethylated cytosines to thymines. And so then you can map to the genome, whatever genome you're mapping to, and then observe methylated or unmethylated cytosines and get your output this way. However, it's been really well categorized or like observed that this is a pretty harsh protocol on the DNA, so it can create fragmentation and it can create damage that can bias downstream analyses. So enzymatic methylation sequencing is a more recent technology, and so this uses an enzymatic over a chemical conversion to convert unmethylated cytosines to thymines. This has you can read the stats, but slightly better, like lower duplication rates. It needs less input DNA. It can actually use the twist panel in particular, can use very low amounts of input DNA, and then better between replicate correlations. And so Twist Biosciences several years ago put out a protocol creating a capture panel of about 500,000 probes that capture approximately 4 million CpG sites across the genome. And so they designed this where it overlaps almost exactly with the EPIC version 2 array. So there is really nice compatibility if you have done studies previously with the EPIC array, or you want to, most human studies are in the EPIC array, even though it's very expensive. So for comparison's sake, the Twist panel is really, really useful. And then that was already kind of out of the box cheaper and did 4 times the number of CpG sites. And then the BIC lab, the Leia lab at Vanderbilt, the Chi lab, and then the Vantage Sequencing Core led a big effort a couple years ago to really miniaturize and optimize this. And so now Vantage offers this protocol at a very, very low cost. Yeah.
I think has a lot of application props. So for the Twist, for these application props.
Sorry, can you repeat the first part?
So for the EPIC version,
oh yeah,
have a lot of replication props to make sure they have measured the accurately. So all the Twist deal with like this. So they use like the reverse trend or forward trend or.
Yeah, this is paired sequencing. So it's pretty, like, we've observed that it's very robust, and even at low coverage, I'm happy to show some stats on that later. But Yeah, the EPIC, that's a really good point, and we'll actually get into this later, but the EPIC array, it's pretty high cost, but you do get very deep sequencing at each site. Twist is paired end sequencing, and it is, I mean, coverage rate is dependent on the number of reads that you're throwing at it, but we have not observed that that's at like, you do a kind of a standard coverage filter, and then we've actually observed that very like extremely high correlation between the EPIC array and twist sequencing for the same probe. So yeah,
Yeah, if you look at the paper, they have a nice like heat map scatter plot that shows a correlation. I guess the replication probes, I guess makes sense if you're doing similar to a genotyping array. If you're doing probes, you want to make sure that it works correctly. But since you're sequencing, I guess the depth of coverage is kind of like a replication approach. So because you're sequencing and you can just you spray painted the genome. If it's protected from spray paint, it's methylated. If it's not, then it is. You can sort of just see how many reads you have that support it being methylated. And
Yeah.
I think it's sort of a difference in how the methylation is being ascertained.
Yeah.
And I guess because the spray paint is less toxic to the genome, then you also get much more like you can get a much more dynamic range rather than just fully methylated or fully unmethylated. You get much more like middle, which is exactly what you want for power for discovering things. You know, things are not just turned on or off. There's some fraction that are and then some fraction that are not. So you also get a much better like ability to see fraction methylation. But it has a good head-to-head comparison in that paper.
Yeah.
Which is
Yeah.
worth checking.
Yeah, that paper exactly goes really into depth on comparing RBS, the EPIC array, and then the twist methylation panel. So I'm just going to explain briefly kind of what the output, like if you were to take samples to Vantage and give them DNA and then have these samples sequenced, what the output is that you would be getting. So the raw sequencing format is going to be a FASTQ. This is something that you should keep in permanent storage. This should be backed up, like doubly, triply backed up. That is irreplaceable information. So if you're going to process from a FASTQ file, right, this is a non-human readable file, we're going to trim off adapters and then map to whatever genome we're using, probably the human genome in this audience. And then that would result in a BAM. And we're happy to like, I don't think it's necessary for what most people are doing, but we're happy to share the code for how to do this. But it's like through standard pipelines, like trim adapt or trim galore, either one. And then a BAM is essentially the reads that are mapped to the genome. These are very, very large files typically. So there are some reasons to keep BAM files if you have a massive experiment. I don't know. I think that takes some it takes some consideration because it is technically an intermediate file type because you can remake this from a FASTQ. It just takes a little bit of time. But if you were going to process a BAM, you would use Bismarck methylation extractor is probably the most common software to use. You're going to extract counts from that. And then BAMs are also useful because they've been mapped to the genome. So if you want to get genetic information, if you want to perform SNP calling, you would use a BAM. The thing to keep in mind with methylation, so we've gotten genotypes from the 3 populations that I showed in the beginning slide, which are indigenous populations around the world. So we can recover genotypes. The thing to keep in mind with doing that from methylation sequencing is that, of course, you've disrupted cytosines. So there are many sites throughout the genome where you won't be able to get the correct base. So that's always something to keep in mind. You can get population structure really, really well, but if you're going to use this specifically for like an MEQTL analysis, you're going to be missing some parts of the genome. But if you don't want to pay for whole genome sequencing, then that can be a good way to go. So I'm happy to kind of chat about genotyping from these files.
If you're using the data from VGI, thankfully pharma paid for the whole genome sequencing. So we can do UTL stuff. Since most of the methylation sample that VGI is planning to generate are the people who are in AGD 250K. So we have genome sequencing, which is great. Yeah.
And then what you'll probably get from Vantage, or I think should get from Vantage, I don't know if it costs like a little bit extra, but is this CX file. It is pretty straightforward to make these. It's like a handful of scripts. And then what this is going to be very, very clearly laid out methylated and unmethylated counts that have been extracted from the BAM for every site in the genome of that particular sample. So it's per sample, and we'll show you that in a second. And then what you're going to do is limit that to CPG or CG methylation. So it's also going to have CHH, excuse me, methylation. And then what we do, and there's another slide that goes more in depth on this, is we just limit to the targeted methylation. So the sites that are actually on the twist probe, there's going to be a lot of off-target sites, but those are going to be sparse across all of your samples. So we just target to those. It ends up being just over 4 million sites because we're adding a little bit of space on the other side, like on the sides of each probe. And then you can take all of those files across your experiment, merge them. So you have a counts matrix per sample, and then I'll get into the additional quality control. So this is where most people are going to start, is at this CX file. And these are so awesome and easy to work with. You can see here, this is CHH methylation. So that is, we're going to filter that out. That was a awesome, nice thing.
I made the slide.
Yeah.
Teamwork,
Perfect.
yeah.
Oh, then you're gonna be left with CG methylation. And then we're just gonna take essentially the methylated, we're gonna extract all the methylated counts. This is a file for one sample. You're just gonna concatenate that across all of your samples, combining the sites appropriately. And so then you're gonna end up with a giant matrix of all of your CPG sites as rows and all of your samples as columns, a methylated counts matrix and an unmethylated counts matrix. We can make all these slides available, so don't feel like you need to be taking notes on the processing pipeline, but this is typically some of the QC that Yash and I think about when we process these samples. So at the sample level, we want to retain high quality samples. Things you want to look out for are how well did they map? If you start with the CX files from Vantage, you'll get a report that has the mapping percent. Just plot it, make sure it looks good. Everything I would say above usually 85%, 90% should be bang on with the sequencing most of us will be doing. Same with coverage. Want to keep high coverage samples. So samples with a mean coverage, you'll see what the distribution looks like in your sample. Also ones with a critical mass of reads, I would check the duplicate percent as well. Most of this will not be a problem for most experiments that you do. If you're really working with low quality DNA, it might be something to pay extra attention to, or really, really small input volumes. And then I also would check the missingness. So just make sure that that sample is very complete. Maybe a CX file didn't convert or something, or it just missed it. We were missing a chunk of chromosome 1. So I just plot missingness at a per sample basis and make sure that I have a lot of data for every sample. And then you can perform a sex check. This is really easy. There's software to do this, but another really easy way to do this is just look at the proportion of reads mapping to the Y chromosome, make sure that that fits with the metadata as you expect it to. And then we also look at the percent of CHH methylation. We want that to be very low, which means that there was a good conversion in the enzymatic conversion step. And then at the CPG level, so now we're thinking about rows, this is what we usually do is do a mean coverage filter. So take mean coverage of a single CPG site. And then for the data that it kind of depends on how many reads you gave to an experiment. But typically I would say minimum of no less than 5 on average across 75% of samples. That's pretty low. And your data will probably be much higher than that. And I'm happy to provide sometimes reviewers in particular can be very picky about methylation from sequencing-based methylation technologies. But we have done a lot of work to downsample and check coverage. So if that's something that is ever an issue for you, feel free to get in touch, and I'm happy to provide some support on that front. Yeah, like I said before, we limit to CPGs on the twist manifest, and then also again remove sites with high missingness. So if you have a whole plate of samples that are just missing a site, amazing, take them out. And then run a PCA on the proportion of methylation. So here you have your methylated and your unmethylated matrices. Add those together. That's a total counts matrix. And then you can take the methylated over the total counts to get a percent methylation matrix, run a PCA on that, and then check PC1, PC2, PC3 across your technical effects. And then typically what we're controlling for is mean coverage, or number of total reads, things like that. But I know some of this is a little bit fast, but we're happy to break down any of it. So essentially what you're working with, the products that you're going to most frequently be working with are methylation matrices. These are, again, the methylated counts, and then you combine that with the unmethylated counts matrix, that's your total counts matrix. These, for a lot of the work that I do, this is like my home base. This preserves the maximum amount of information. So if you're doing modeling to understand how methylation is associated with age, for example, you can do that in a binomial model. And then you're taking into account the confidence or the read depth because you are inputting two matrices, your methylation and your total counts matrix, because 50% methylation could be from 2/4 reads, or it could be from 200/400 reads. So that's information that's kind of lost if you collapse into a percent methylation matrix. However, there's a lot of reason to work with percent methylation matrices. These are really akin to what comes out of a microarray. So the epic array works off of beta values, which are scaled 0 to 1. This is essentially a percent of methylation. So that is the closest approximation between the technologies. And a lot of out-of-the-box software that you're going to be using if you want to employ like a previously generated epigenetic clock, it's all based on percent methylation. So there's a lot of reasons to have kind of these, I would say these three matrices. Also cell type proportion estimations, generally, I mean, almost always actually going to be based off of percent methylation. This also is really easy to input into a linear modeling framework because you just have one vector of numbers for that given CPG site. Okay, that was like a torrent of information. Yeah.
So, sorry if I missed something, but for the cell type of Pershad, we need to perform a decomposition for this button. Is there already a reference panel for the sequence-based methylation, or is there any kind of reference-free method?
Yeah, but it'll be in the next section, but
Yeah.
it's essentially there's all of it has been built off of the arrays. So Hannah, who's right here, is working on making one for that enzymatic methylation arrays, but they work okay on the ones from the arrays because the CPGs are somewhat correlated. They work okay. So we've been using the ones that are currently exist, but nothing exists yet to date on the enzymatic methylation.
Yeah, but comparing the estimations from some of the common softwares, it does a decent job with like a
Which actually, yeah, show it later. It's okay. So that's reasonable. And I guess the thing that I'll add just about this is like basically the first bullet is unique to sequencing based methylation. So basically, if you were doing an epic array, you'd only be using this entirely, and you basically have no control over how many reads you're getting of a particular site because it's very standard. So I think that's why it's really nice to actually just keep the raw counts, as Marina mentioned, for these kind of ideas where you might not get perfect coverage at a particular site.
Yeah, exactly.
Yeah.
For example, we're making epigenetic clocks in our lab, and you can do that with like all of the CPG sites, or you could limit it to ones that are really, really high coverage. There's reasons where you might want to do one or the other. But this is, I think, really important information that you do get from sequencing-based technologies, which is that read depth. And then the thing that Yash kind of alluded to earlier that I'll mention now is that when you're thinking about downstream analyses, there are times that we want to use what's called variable methylation. And then there's times that you might want to include invariant methylation. I don't know if we have a plot in the next couple of slides, but we do. I guess I'll just drop it. So if this is 0% methylation and this is 100% methylation, we could have a CPG site that across all of our samples has basically 0% methylation, and most of the genome is either going to be extremely lowly methylated or extremely highly methylated. So typically we think about variable methylation as being between 10 and 90%. But if you have a lot of power, like if you're working with a lot of samples, you can kick that out to like 5 to 95%. But as Yash mentioned earlier, if you're working with like 2% methylation on average across your samples, and then you're trying to model an age effect, there's just such little variation to work with. You're probably not going to see anything. So we kick it out because you're going to have to correct for that in multiple hypotheses. correction, and you just lose power. So this is an important consideration. However, there are a lot of times where you might want to actually include invariant methylation. A lot of clocks do rely on these invariant sites because they're really, really predictive of stochastic age effects. But that's just a consideration. So if you're modeling like X environmental effect, think about using intermediate levels of methylation. Okay, I think that's
Cool.
Yeah.
Any questions? Any other questions?
Yes.
Cool. Keep going. Sounds good. So I guess I kind of talked about this previously in the setting of EWAS confounding, but why would we care about cell composition? EWAS results can be confounded, and the reason behind this, which I'll show in a next plot, it's just that different cell types in your blood have very different methylation patterns. So the sites that vary between those immune cells as their fractions change will be much differently picked up on when you extract the DNA of all of the nucleated blood cells in your body. So that's interesting. And then it's useful to infer cell composition from only DNA. It's especially useful if you don't have as much phenotypic data correlated with the same sample. So you could then understand the cell composition from the DNA alone. That's less of a problem in BioView because most of the DNA is extracted from CVC tubes. So that gives you a complete blood count if you have access to the synthetic derivative. But both of those are useful. I think that there are many other reasons, but those are sort of two that came to mind. So this slide is from Hannah. She's working on this really important problem. So basically, how cell deconvolution works is basically you have a reference signature set of what you know a granulocyte looks like, what you know a lymphocyte looks like, what you know a monocyte looks like, and then you're sequencing the whole blood, which is a fixed mixture of these known patterns from basically flow sorting and understanding what each particular cell type looks like. So then if you know what each part of the mixture looks like, and you know what your actual mixture looks like, you can then estimate what fraction is being contributed by each cell type and what fraction of the immune cells are of each fraction. So that's kind of the idea behind cell proportion estimation. So then it would tell you, based off of this, the most probable most likely explanation is that 50% of your blood were granulocytes, 30% were lymphocytes, and 20% were monocytes, and we observed a mixture of all of these together. There are models that are I would say, very new and very, they need to be improved, but in theory, you can take a bulk methylation sample similar to a bulk RNA-seq sample, and you can infer the methylation of each cell proportion. So you're sort of like flow sorting the cells without actually flow sorting the cells, but it's not very good. So I would caution against using it specifically, but it can be useful if you really, really care about a specific cell type, and you could pair it with data where you have flow sorted it, so you could validate what you found. But either way, number one is what we really need to do epigenome-wide association studies. So we'll focus on that. Here's like an example output of cell proportions. So this is using methyl CC. We inputted the beta matrix from at particular sites that methyl CC uses as its reference site, which is about, I think, 500. And then it'll output you these fractions of what is the composition of your sample, basically what fraction of your DNA is from each cell type it's estimating.
And if I can just jump
Yeah,
in
go
here.
ahead.
These softwares are like, there's two methyl CC and then EpiDish that we typically use. These are like, it's like one line of code. So these are really, really straightforward and fast to use, and the output's really quick. So that's just
Yeah, and I provided some code that we use to run this. So yeah, then there are many methods. I talked about methyl CC briefly. People have benchmarked this. So this reference in Nature Communications last year, and then another one came out recently. Where like, how do these perform compared to like each other? Which one should you use? They all kind of are okay. Honestly, I don't know what the accuracy score means. I assume it would be from 0 to 100%, but it's 0 to 500. So I'm not sure what the accuracy score means, but you can kind of see that they're all around meh. And I think it's a great question. You might expect different performance with enzymatic sequencing to ascertain methylation compared to epic arrays. So, and that's not been fully benchmarked yet, but Hannah's working on it. But so for now, we kind of have to live with what we have. And then this is using BioView data. We have CBCs from the same blood draw that contribute the DNA to the sample. And this was, I think, among 500 people that we had sequenced, the big lab had sequenced in BioView, and we see that it's far from perfect. So the dashed line is y equals x, and then I combined all the lymphocyte cells, so NK cells, B cells, CD8, CD4 T cells all together, and you see that they're correlated, but not well at all. And then the monocytes are particularly kind of concerning. I think this is confounded though by BioView lets the DNA sit for about 1 or 2 days in the fridge before they sequence the DNA. So it's possible that different cells than were in the CBC made it into the were extracted for their DNA. So I think it's hard to know exactly how well they work, but I guess this is just to be cautious about the output. Don't sort of stake everything on the output of the cell proportions, but it's still useful. You probably want to put it as a covariate in your model.
Yeah, and we've observed the same thing across the kind of multi-population data set that I'm working with, where again, monocytes compared to a CBC don't really perform well. This is like problem Hannah's working on. So if you have a monocyte specific phenotype, might want to think about if doing some isolation beforehand is more appropriate than doing something in bulk. But otherwise, because there's a much smaller percent, we find that this is very comparable to using like CBC counts. So yeah.
And I guess it doesn't mean that the methylation of the monocytes is wrong. It just
Exactly.
means that you're not able to estimate the fraction of the cells that are monocytes, just to clarify that. So it's not like you're missing the methylation of those cells. So how has this been used by our group? I sort of mentioned it in the EWAS context, which we'll get into, but Alyssa Parker, who defended, I guess, last year from our lab, in terms of clonal hematopoiesis, which is what we study, we're very interested in seeing how the mutation size in your DNA changes over time. And since the mutation is preferentially harbored by certain cell types, knowing from just the DNA how many how many cells of what type are in your blood actually allows you to adjust for the cell types and seeing where the mutation is harbored. So this is kind of like a somewhat like clinical translational application of using methylation to estimate cell types to improve, somatic variant interpretation. And we can sort of see like whether your clone was shrinking, growing, or stagnant sort of changed if you actually paid attention to the cell proportions, which was a cool result. Cool. Any questions about cell proportions and deconvolution? All right. Maybe, yeah, everyone's waiting for the EWAS part. So yeah. So basically this is now going to be your EWAS section. There should be an arrow connecting cell proportions to EWAS, sorry. And we can dive in. So what you mentioned this, in a lot of you mentioned this, that you're interested in doing EWAS and connecting environmental exposures to and understanding their association with the methylome. So what an EWAS is, it just tests for the association between DNA methylation and a phenotype of interest. Since we're the Genetics Institute, it's like a GWAS, but for the methylome. And I think there are three key concepts that I wanted to mention for EWAS. Number one, for this use case, I haven't actually implemented any of the binomial modeling, but that would be cool to try. But for this use case, I've only talked about beta values. So this is a 0 to 1 value representing the fraction of reads that are methylated. Similar to a GWAS, it will require multiple test correction because you're testing, hundreds of thousands, if not millions of CpGs. And then cell type composition is a critical confounder. So basically the pipeline, which I'm showing on the right, is just you have samples with their phenotype data, their metadata. You perform methylation profiling, and then you do a statistical test to get the per CpG region. What's the association with the phenotype? And then you'll get some summary statistics, which hopefully will lead you to some insights.
I'll just jump in quickly. In addition to controlling for the major cell type proportions, so typically lymphocytes and neutrophils, I would also we should also include technical effects on here. So from that PCA of proportion methylation, we often include it's usually between 2 or 3 technical effects. That's incredibly important if you're combining across batches. So obviously, if you're know about sequencing, batch effect correction can be quite challenging. So I would just a strong word of caution, obviously for any study, but particularly for sequencing-based technologies.
How do you correct for it? I guess you just encode the batch as a dummy variable or something.
Yeah, well, that is why we
Yeah.
tried to put everything on a single batch. Just it is sometimes almost just be very careful with randomization, I guess, and experimental design is because otherwise you can end up almost being unable to do comparisons. So it just takes a little extra thought. If you're like, I want to get this off to the sequencer, we're getting another another shipment in a month. I don't know. If it was me, I'd probably just wait. But just a
Yeah,
word
that's a
of
good
warning.
point.
And then, yeah, for all of this, we're talking about modeling at the single CPG site level, or well, I guess you'll get into that.
Yeah.
But typically, yeah.
Yep. So yeah, I guess if there's sort of two ways of thinking about this, which is fundamentally, I think, different than germline genetics vs. GWAS. So you could either treat methylation as the exposure, and this is sort of stuff that we've done where, you know, if someone has a disease, if someone has cardiovascular disease, what DNA methylation associates with having disease. So the methylation is predicting your exposure. And I think many of you talked about this as of interest to you. What's the methylation signature of breast cancer, for example? And in this case, you know, you could have an outcome. I talked about disease, but you could also have a continuous trait like lab values, anything like that. I won't tell you how to design your research project, but just so you know that you can use it as exposure. And then you could also use methylation as an outcome. So does a trait alter DNA methylation? So you then can have DNA methylation on the left side of the squiggly line in the linear model, and then you have an exposure. And this is sort of a way to capture the environmental effects of on DNA methylation. And I think it's particularly powerful if you're able to do both in conjunction, because then you know you can then use methylation as a mediator or a connector between the exposure and an outcome, which I think a lot of you seem to be interested in. So that's sort of just to make sure, at least for me, this was an adjustment because I was used to the SNP always predicting the outcome in germline genetics. So then, yeah, we kind of alluded to this. There are basically three ways to represent the methylation data in these models, and they all have their pros and cons, which I tried to summarize here. So negatives are negative, positives are good things. Blue is kind of like neutral. So you can either, so you could do it kind of like a GWAS where you do each CPG is predicting your outcome or altered by your exposure. And then you're going to basically do then, you know, let's say you pick 850,000 because you are merging your study with your old EPIC array data. So you picked 850,000 tests to run. This is difficult to interpret, to be completely honest, and it's not much different than a SNP in the genome where, you know, what gene does that map to? What is the function of that SNP? How does it affect, you know, does it affect expression? Is it in an intergenic region that means nothing? So interpretability can be a problem when you have these single CPGs, because if you wanted to do some kind of pathway analysis, you have to map that to a gene. You have no idea what that CPG sort of does in the cell type or context that you're interested in. You're also having issues with multiple testing penalties there because you're testing a lot of CPGs. So for something to be significant, assuming you use Bonferroni correction, or it would be 0.05 divided by 850,000 would be your alpha. So you're assuming that you can use, you know, other FDR correction methods, but this also puts you at high risk of false positives, I think, because you're testing so many things, they could be associated. But you'll not be at risk of false positives as much because you're going to correct for multiple testing correction. And then this, because of the number of tests you're doing, you may miss out on association. The second approach is this sliding window approach. I think, you know, people can argue about the optimal genomic window size. I just picked a random 2KB in this case, but you can basically march across the epigenome, and create windows and then take the average of the CPGs in that window. And, you know, people tend to have overlapping windows. And then basically you then know from, instead of having your, you know, the rows of your beta matrix be chromosome 1, position 10, which is a cytosine, instead it would be chromosome 1, position 10 to position 2010. And the methylation fraction that you get is actually the median or mean of all of the CPGs within that window, which can also be a variable number of CPGs because we know that it's not you know, equally spaced across the genome. There are sections that have lots of CPGs and sections that don't. So this is a little bit more interpretable because you can probably, because you can map a region and say, oh, that's in the promoter of this gene, for example. But the window size that you pick actually matters a lot in that case then, because the issue is that you're basically going to create, yeah, I guess false negative should have two negatives on it, not like you're at very high risk of false negatives because you're collapsing across CPGs that may be completely uncorrelated with each other. So then you will then dilute your signal and you'll find nothing, especially if you pick a window that's very large and you could have a biologic CPG island that's much smaller than 2 kb, but now you're lumping in all of these invariant and random CPGs along with it. So that's a risk.
I'll just kind of add to this. This is where methylation data gets like really, really fun, in my opinion. And I typically work in the space of column 1, the CPG by CPG approach. Is it fair to say you typically work in column 2? Cool.
Yeah. Yeah, I think that, yeah. Or I like to do both, but yeah, that's right.
And so a few considerations here that Yash touched on is you can either make, if you're going to combine, there's a lot of biological reasons, right, to combine or aggregate methylation in certain areas of the genome. Either they're biologically informed regions or because we're trying to look at the clumping of CPG sites, because a single CPG site on its own is probably not going to be that influential in gene expression. So you can do that on the front end, like Yash was saying, sliding window approach. You can also do that on the back end. So if you take a CPG by CPG approach, there's reasons why you would, instead of defining your regions a priori, where you would define them via permutation or something on the back end. So you could ask, like, if I have an age effect, is that more likely to be like clumping together? And you can ask that by permuting your p-values. So there's kind of different biological considerations and ways to parse the data depending on exactly what your question is. So there's also the consideration of, like, it's always good and really powerful, I think, for sequencing-based technology to keep in mind that here, because we have the number of reads, this is such an advantage over, in my opinion, the EPIC platform. If you're trying to aggregate methylation, you combine all of those reads, which gives you a lot more power, because that's very different. It's very different to say, like, these 10 CPG sites, some might have a coverage of 5 reads, and the next one might have a coverage of 50 reads. That's different information. Those might both be 50% methylated. So if you're going to, well, I guess in that case, let's say the first one's 20% methylated and the second one is 60% methylated. If you collapse that and you have the EPIC array or something, you're just averaging, but that's not totally correct, because we actually have a different depth of sequencing, so different information. So you can then aggregate, arguably, I think, much better to see the methylation in a particular region. So those are just some of the things to keep in mind when you're thinking about exactly how you want to parse methylation data and model it.
Which is basically like a reason to use the first bullet point.
Yeah.
Because then you actually have the count. So what I said was, applicable to the arrays, but instead it actually is better to take the sum of the methylated reads in that region divided by the sum of all the reads in that region.
Yeah,
Because
exactly.
then you would not get 40, you would get closer to 60, I think, is an example. Agreed, 100%. Yeah, the third column, so maybe the optimal approach is a mix between column 1 and column 2, which I think is what we were talking about, which is basically you could do an EWA CPG by CPG and then understand which CPGs are associated with your outcome and then lump them in a way that's sort of useful and interesting. This is, I guess, you don't want to double dip and make these fake ones that will be associated, but you can like plot the CPGs in a particular region that you're interested in and see how certain CPGs associate. And that's like a common visualization that people use for the CPG by CPG approach. The final approach is sort of like taking ENCODE or basically like annotated genomes to say that we know that all of these are in the enhancer of this gene. We're going to lump them all together. I think I put it as an option. I find it to be really difficult to justify the choices that you're making, and difficult to pick which cell, because all of the ENCODE tracks are by tissue or by cell type. So then it's kind of like, what is, obviously you could use the whole blood one, but is that really what you want to do? Is that connected to the question that you're asking? If you're looking at, some other disease of interest that's not directly related to the blood. So I think it's just hard to justify what you're using. And then a lot of them will be very sort of, it's very curated, which is good and bad. So I think that can be, I've had little success with the third option, to be completely honest.
Yeah, and I'll say here, this is going to, if you're limiting yourself to particular areas of the genome that might be functionally relevant, sure, that is going to throw out a lot of information because most of the genome is not functionally relevant or a promoter or enhancer. The reason I think you would want to do this, or like, is if you had especially paired gene expression data. You can do it right with gene expression data that's not totally paired from the same population, or otherwise. But if you had paired gene expression data from the same sample, and you're looking at promoter, methylation or enhancer methylation and associating that with genes, that's a really great reason to define that promoter 2 kb up from a transcription start site, and then ask if the methylation in that region predicts gene expression.
Because you
So-
want to map it to a gene in that case, very
Exactly, yeah.
strongly, very predictably. Do
Yep.
you have a question? Yeah.
So like for the CPG, it's especially bad for like for that, like because you're dealing with like swirlings of CPG sites. So I guess then to get past the full testing penalty, like
Yeah.
whenever you're talking about doing CPG by CPG, then like doing a larger area, so are you just looking at something with like maybe taking like your lowest key value places and consolidating to those, then doing like the sliding window, or what was your?
Yeah, okay. So I'll break that down. This is a really good question. So with this panel in particular, I'll just describe quickly the data set that I've primarily been working with. It's 4,000 samples across 3 different populations around the world. So there's very different genetic backgrounds and environmental backgrounds, these populations. And we have taken, so there's 4,000 or 4 million, excuse me, CPG sites after all of our filtering that we laid out before. And then we're asking CPG by CPG, is there an age association? Controlling for cell type, controlling for technical effects, controlling for population structure, et cetera. Of that, let's say that we get 2 million that are significantly age associated in one of the 3 populations. So that goes through a very rigorous multiple hypothesis testing, or like FDR correction. So they're already at that number of CPG sites, I'm looking at like a 1% FDR threshold. What we've used, there's a couple of things that you can do to recover some of that information and the power. One thing that we've been using that's if you have any like cross population or cross condition, an experimental design that is something like probably a cross condition for a lot of folks in this room, I would think about using MASH-R, which employs effect size sharing. So you're using a very, very low false discovery rate threshold. So you're being very stringent, but you're sharing information about the structure of the resulting data to ask, are the effects between, in this case, populations? So a single CPG site, does that have an age effect in population A? Is that age effect, maybe it's not significant in population B, but it's pretty close. So then the program is using that information to say, yes, indeed, this is an age effect across the populations. And so there are some techniques, MASH-R is probably the most useful, or like the most widely used one, to rescue some of that extreme false discovery rate.
It's kind of like, I guess, a mix between a meta-analysis and a like multi-trait
Yeah,
GWAS,
that's fair.
I guess.
Yeah. Mhm.
Yeah.
So
But like you could think about if in BioV you had samples, you could look at diabetes, cardiovascular disease, obesity, and then share across.
Share
and then
across,
see
that's a
like
good example.
final stuff.
Yeah.
Um,
So a lot of methylation work just comes down to defining the question. And if you ever have questions about like that, we're happy to talk strategy on how to model.
Cool. So we kind of talked about this, but I'll explicitly state it again. So you'll have a beta or coverage matrix with rows being your CPGs. If you were, if you had epic array data and enzymatic methylation data, it's important to remember, I guess, the CG IDs that you may have worked with in the past they're made by Epic to interpret their probes. It's like a probe ID. So there will not be any CG IDs on the data output from the sequencing, because that you just have a position that is a cytosine that has a fraction methylation. So you can map between enzymatic methylation and epic array easily, and you can convert that to a probe ID if you really want to, but how we leave it is mostly in terms of chromosome and position. And then you'll have sample IDs as your columns, and then the actual values, we talked about it being from 0 to 1. And then similar to a GWAS, the information from one CPG, if you're doing it, is not relevant to doing the regression for the next CPG. So you can easily chunk your approaches and parallelize it if you'd like to make the EWAS run faster. So each regression is somewhat independent. So then you can leverage that to, make it run faster. And then how I've set it up is just you have a phenotype metadata file, which has the outcome variable of interest, let's say case control status. You'll have a sample ID that's shared in the same as the column of the beta matrix, and then you'll have the sample level data that you want to adjust for. And then I did not use a genetic relatedness matrix, but it's really relevant depending on your question and your population. So we wanted to talk about that.
Yeah, so for the data set that I described, right, we're working across three really, really genetically diverse populations. So here, not only controlling for like the population explicitly, but also we've created like a combined genetic relatedness matrix that controls for fine-grained population structure. So if you're using something like BioVU, it might be totally reasonable to just put an ethnicity as like a covariate column. But if you're working like with family groups, let's say, 'cause we've Turkana pastoralists, and there might be multiple people from a family or family groups in a particular region that we don't know that they're family groups. In that case, we can actually recover genetic relatedness very well. So there's that limitation that I told you about before for doing MEQTL analyses. But if you're just trying to recover population structure and understand, yeah, like kind of fine-grained structure between different ethnic groups, does really, really well. You can do that with actually not that many sites. Yeah. And I can show you plots. I don't think I put any in here, but yeah.
Awesome. Yep. And then we had cell proportions we got from EthylCC, EpiDish, whatever you want to use. You'll have your age, sex, batch specs, PCs. We can use that especially because we have biovehicle genome sequencing data. Rather than just having ancestry, you can put the genetic principal components if you wanted. And then it'll output an EWAS summary statistics file, which has the effect size, the beta or the odds ratio, a confidence interval, and a p-value. So just I wanted to show you actual examples because this tends to stick better in my brain. So for example, if you had an array and you combined your data with some array data and then tested with your probe ID, you would have these CG numbers. If you use the enzymatic sequencing alone, you would have chromosome 1 position 10, like I said, and then you have samples as the columns, fractions in the rows, and then your metadata / outcome file are on the right. And then the cool part about this is that you can actually do any type of regression. So depending on your problem, you could do a logistic regression to test for whether or not they have the disease or something. You can do a linear regression if you want to see the age association, or you can do time to event, which would be particularly relevant in a disease cohort, if you're looking at a disease in BioView. So here's basically the pseudocode for the WDL that I'm providing to you guys to run CPG by CPG EWAS. So basically you'll have chunks of your beta matrix, which are basically regions of the genome that I've put into a single matrix to then run them in parallel. The metadata file for each sample, the outcome and the covariates, and then what it will do is just scatter all of the chunks in parallel, and then for each CPG in that chunk, do a logistic regression to see how the CPG is predicting your outcome, and then it'll store the odds ratio, 95% confidence interval, and p-value of the beta coefficient of that CPG. And then at the end of it, it will gather all of the data, combine it into a single summary statistics file. So it has options to do logistic regression for a binary outcome, linear regression for a continuous outcome. I haven't implemented Cox regression, but you easily could. And then parallelization is implemented, and then you can set the chunk size. I did it as 5,000. Depends on how impatient your boss is kind of how I like to think about it. But theoretically, you could spin up millions of computers, and it would take you the time it would take to do one regression. I don't know why you do that, but you could. Yeah, if you really wanted it instantaneously. For loop across all of the CPGs. And we talked about the output. Here's an example of the sliding window differentially methylated region EWAS, which is you have a beta matrix, a window size, and a step size. So you'll note again that they're overlapping. The window size is 2,000, and basically 50% of the windows will overlap with the next one. And then you can set a minimum number of CPGs that you want to include in that window, and then you can either take the median or the mean across the CPGs. You can also, yeah, I guess in this case, I did not leverage the first bullet point that Marina mentioned, so I should fix that. But this is just collapsing it across the CPG's fractions because that didn't keep the count matrices. And then it'll tile the genome, and then for each of the windows, it'll do the same thing as if each window is a CPG, kind of analogously. And then, yeah, this is just like a idea of what the summary statistics will look like, very similar to a GWAS if you're familiar with that. You'll have a coefficient, you'll have a standard error, you'll have a p-value, and then you could do a FDR corrected p-value, and then oftentimes this pipeline won't do it, but you can then take UCSC or other annotation approaches and map the site in the genome to a gene and then look at that in that way, which is kind of like the post hoc filtering way to think about connecting it to genes rather than collapsing a priori. So yeah, then I think this is sort of similar. If you're familiar with regression, it's kind of not that groundbreaking, but you'll get a beta regression coefficient, which is basically the change in the outcome unit per increase in methylation beta. So then let's say if you have a negative 0.1 beta, then a 10 percentage point increase in methylation will decrease your outcome by 0.1 units, let's say, for linear regression. And then for logistic regression, if you have an odds ratio of 80, then a 20% lower odds of your outcome per unit increase in methylation. I think this is important because if you're actually looking at what is biologically meaningful, commonly people will look at a 5% methylation between your groups if you have a categorical outcome. So if you you can sometimes have results where you can say with very, very large statistical certainty that the beta value will change by 1% between your cases and your controls, but because you have large sample size. But is that actually meaningful biologically? Probably not. And then, yeah, so there's no consensus threshold, but just think about the beta is basically kind of my point. What threshold do you want to count as biologically meaningful? So you don't want to only filter on p-value, you also want to filter on effect size. So how can you visualize your results? We don't really need to go into this, but you can create a Manhattan plot. You probably want to examine the QQ plot for inflation to see if you have spurious results. And then you can make a volcano plot, which will allow you to visualize the odds ratio or the beta on the x-axis. So these are two examples of two ones that I've made.
The other thing I'll just
Yeah.
throw in here, it's really fun to actually be able to do this talk with you because we approach things from like pretty much the same but slightly different reasons.
Yep.
First thing I do when I do the binomial model is just a histogram of the p-values, and you're just looking for a non-uniform distribution, just like really low. If you line up all the p-values in a histogram, this is count, and these are your p-values. Uniform, there's nothing here. If you have low p-values, great. Now we've got a signal to work with.
Right, which is analogous to on a QQ plot, you have dots above y equals x. Yeah.
And it's like the EWAS, just instead of stratifying by the genomic position, you're just doing it by p-value.
Yep, cool. Yeah, and we've applied this. So the left one, I just wanted to show how flexible this can be, which is cool. So the left one is actually using methylation as an outcome of adversity exposure. So housing insecurity, financial insecurity. So the CPG was the outcome. The right one is how do CPGs predict whether or not you have clonal hematopoiesis. So that was, you can do either way. Cool. So yeah, now maybe the most interesting part. How can we actually like learn from this random set of matrices that we've created and results?
Are there questions on the
Yeah, sure.
modeling setup? Because Yash and I kind of approach this from again, like slightly different but very complementary angles. So there are, it's pretty flexible in terms of what you can do. Okay.
Do you feel like you could EWAS your trait of interest, breast cancer? Ready to EWAS?
Yeah.
Okay. Oh, yeah. Okay. Cool.
Cool. Okay, sweet. So now we have our results. So we have our, let's say, beta value from our, we'll pretend linear models in this case, p-values, we've done an FDR correction. And so now we have our sites, our CPG sites of interest or our regions of interest. And I also, if anyone's interested in kind of how you might chunk or do region, those permutations, the region-based permutations that I said on the back end, I'm happy to talk about that. I don't think it's pertinent right now, but if you're trying to like understand biologically relevant clumping of your effects, chat with me if you're interested in that on the back end of things. Okay, so yeah. Oh, that's a great question. So essentially, like, how much missingness can we handle? This is another thing that I really love about the twist methylation panel. Before this, I worked with RBS data a lot, and the missingness was just like super frustrating. It was very hard to deal with. This panel is very complete, which is awesome. So the linear models or whatever models you run obviously will kick out any missing sites. However, that should be a very, very small percentage of sites across all of your samples for any given CPG in the twist methylation file. That is a quality control metric I would look at, is just, do you have a CPG site with really high missingness, like over 10% missingness across your samples? I would probably go ahead and throw that out, but you'll look at the distribution. So in terms of that, you'll lose any sites that are missing, but again, because of how complete this is, I would just impute any missing data. You've limited it to a very, very small amount of missing values across your CPGs and your samples. I'd go ahead and impute. Then you just have a complete. That can take a lot of computational power depending on your sample size and the CPG set that you have. But that's what you're going to use for clocks, for cell. Like it's the most flexible to have complete data, and it's been shown, I mean, just repeatedly, that we do a pretty good job if we have a big enough sample size. So I usually do not do that when I model. I use that for like clocks, but I'll just throw out, the first line will just be filtering for missingness, but it again depends what you want and what the data look like. So
Yeah, you can sort of like falsely introduce missingness into your data by
Mhm.
being too stringent in your filtering
Yeah.
upstream, which I've made the mistake of doing before. But so I guess because similarly, because it's sequencing, let's say you only want to pick sites that have let's say you set a min coverage of like 5x for every site. You don't want for every sample has to have 5x or more coverage at that site, and then 75% of them have to have 15x coverage at the site. Let's say you then take that, you create a matrix. Any person who has coverage less than 5x at a site will be NA in that matrix. But because you have the actual counts, you could then go back and just fill it in with the results that they had, or not do them in coverage filtering initially, and then you'll be able to have a low confidence, but you at least have data there that you've actually measured. And that could be a complementary approach to imputing, because we do know that CPGs are correlated that are nearby. So you could also learn from the CPGs that are nearby that did have coverage, or use the data that you have that's imprecise due to low sampling. But either
they're
way
probably
is,
pretty comparable.
but yeah,
That's a
exactly.
good point.
So either way is good, but you don't want to introduce missingness for no reason, which I've done. So don't do what I did. Yeah.
Okay, sweet. So now let's do some biological interpretation. All right, so typically when we get results of a bunch of CPG sites, like we've been alluding to, it's a little bit hard to make, like how do we interpret like 2 million CPG sites that are associated with age? One of the things that I find very informative, that a lot of, like, it's not a sexy analysis, but is to just understand like where in the genome are they occurring? So are we seeing things that are falling into transcription start sites, into enhancers, or are effects mostly happening in quiescent and heterochromatin regions? This is, particularly useful for age effects. So on the bottom here, this is just showing that the baseline methylation that we expect across the genome differs by genomic context, right? There should be very, very low methylation in promoters, in enhancers. Enhancers are a little more flexible in general. And then in quiescent regions, heterochromatin regions, those are typically going to be really highly methylated. So your data will almost certainly look like this if you were to just get average methylation for a genomic region, but you can use Chrome HMM for your particular, you can port over these annotations for your particular tissue of interest. So they have PBMC annotations that have been done, and this just chunks the genome into it's actually I think it's 16, maybe it's more now, regions.
So like there are options, and like 12 or 16 or something.
Yeah. And they also have, these annotations have been done across a wide variety of tissue types. So this is kind of a nice first stop to say, am I looking at things that might be influential for gene expression? Or am I looking at things that are kind of in like, quote unquote, like junk DNA? I really don't like that term, but like less transcriptionally relevant regions, which can give you pretty good insight on if you should actually expect an effect downstream or not. Okay, the next is the EWAS Atlas. This is a really great resource, especially if you're working with like US hospital samples or kind of US or European samples. because this is an atlas where people have uploaded the results from their prior EWAS studies. So the CPG sites that are associated with particular traits. So let's say that you are looking at heart disease in a particular study. You can go to the EWAS Atlas and download the hits that other people have found around the world for heart disease and say, like, am I on the right track? Is my data matching up? Or do I have a reason to think that we might actually be looking at slightly different sites? And so we can do this overlap for a lot of different reasons. I find the EWAS Atlas slightly clunky, but you can get to the point where you can download it's kind of a pain, but the next is gene annotations. So associating your CPG sites of interest or your regions of interest with either the nearest gene is one approach. I think there's reasons to put like kind of space parameters on that. But a common thing also to do would be to ask from the transcription start site through, or I guess through the end of the gene body through, I don't know, like 10 kb upstream of a gene. You could say, what genes are close to my CPG sites of interest? And then ask, are those enriched for particular biological processes? So these are really easy. Like, you just associate your CPG sites, number of CPG sites with a gene, and then use gene ontology or GSCA. These are really like pretty straightforward softwares, either that have like web browser abilities or can be done in most, just statistical softwares. The other thing is you could maybe ask if there's tissue specific overlap. So if you have CPG sites that you want to understand if they might be specific to X, Y, or Z cell type, you can port over annotations from other studies. Transcription factor binding site enrichment is another one. So again, just thinking about kind of spatially in the genome where you might want to limit your search space to, but then to ask, like, what transcription factors are binding in these CPGs? And then do I see a really highly methylated context? Okay, that's interesting. Maybe that methylation is actually inhibiting the transcription factor binding, or the transcription, is this a lowly methylated context? And I think those transcription factors are able to bind, which can give you some insight for then the genes that those transcription factors are associated with. So just sort of ways to triangulate what those CPG sites might be actually doing, which is sometimes a little like reading tea leaves, but can often get somewhere. I mean, there's so many other annotations, but these are pretty standard. Okay. Do you want to explain variant under probe?
Yeah.
Okay, cool.
There's been a lot of interesting, as you do methylation, sequencing and methylation, study methylation across more diverse samples. It's become an issue with epic array that you can have actually variants that disrupt the binding of the probe, which then alter what methylation you ascertain with, because if the probe doesn't bind, then you don't see that site was methylated or not. This is not a problem with YAMSeq. So I think that's a big advantage, especially if you're applying it to samples, non-European samples, essentially. So I think that's just what the only point I wanted to mention about variant under probe, that there are no probes, so there can be no variants that disrupt the binding of the probe, which is valuable. And people have found that is a contributor to differences in methylation when they study how germline genetics connect with methylation data. But there is real allele specific methylation, which makes sense, where if you have a SNP, let's say you use the whole genome sequencing in BioView and you have methylation on those people, you could perform an association between the methylation fraction at that site and if you have a variant near that site, and that could tell you about the biology and the regulation of that that CPG, which is exciting and cool. And then similarly, the methylation QTLs can provide you with some degree of causal inference. So you can look at what do your genetics predict your methylation at that site should be? How does your methylation at that site compare or contrast with that level? And then is the genetically predicted methylation levels at that site associated with disease or not? Is the observed methylation at that site associated with disease or not? Which could be really, which allows you to understand, is the methylation causal in connecting to the disease, or is it an associative biomarker, let's say, of getting that disease, which are both options as to why it's correlated with your disease from your EWAS. And then, you can, oftentimes people will limit to genetic variants near that CPG, but there are also some regulation that occurs across the entire genome, let alone, or on the same chromosome. So I think there are different ways, different questions that you can use, but all that being said, you can basically connect germline genetics to methylation. I think that's a unique power of our cohort, or BioViews data.
Yeah, there's tons of questions to add, like MEQTL as a whole
whole-
field essentially.
yeah.
So
But I think the top med cohort and some other cohorts across the world have done this kind of study, so that those summary statistics are available. So if you cared about a particular CPG from your study, you could then understand how germline genetics influence that CPG, and then dive into that a little bit more if you'd like. And since you want to use a different cohort than what you're actually measuring it on, so then having that data is a really valuable resource. And it stands for methylation quantitative trait loci. Let's define that, sorry. Yeah.
okay, so epigenetic clocks, a couple people said they were excited about that. That's great. I'm really excited about epigenetic clocks and work with them and to make them a lot. So we've touched on this a little bit, but I just wanted to kind of highlight one major way in which enzymatic methylation sequencing in the twist panel differ from commonly used microarrays like the EPIC array. So microarrays, for reasons that I don't fully understand. I mean, I do understand it, but like essentially because of what they perceive as the confidence at the extremes in their microarray, essentially do an offset towards more intermediate levels of methylation. So they literally just like add a value to either end, which compresses the the estimation of proportion methylation from 100% to more like 95%. And that's fine. It's just it's a feature of microarrays, which I think we recover more realistic levels of methylation at high coverage, obviously like thinking about coverage. But if you got 100% methylation and you have 300 reads on twist panel, like that's probably that. Whereas the EPIC array is going to adjust that value in a little bit, which is fine, but it's something to keep in mind for when you're looking at the correlation. If you want to do a study where you're trying to incorporate both data types and you're like, these are just not lining up, particularly like the extreme values of methylation, you're going to see that it's not falling on the x equals y axis, and it's because it's EPIC array's fault. And so we have done a deep dive on comparisons from paired samples, which is in that Longton 2025 paper that we provide at the end as well. But I've also done a lot of comparisons beyond what's in that paper and there's basically the twist data recapitulate what you can get from EPIC very well. So for example, one use case is just comparing the per sample. So if you had a sample that was done with EPIC and a sample done with twist, the per sample, like across the entire genome, that correlation is across 56 samples that we tested, had paired data for, is over 0.96, Pearson's R correlation coefficient of 0.96. And then if you did the EpiDish deconvolution approach that we alluded to earlier, extremely high correlation in the estimates that you're getting. So I've noticed hesitancy in a lot of the human genomics world to move beyond the EPIC array, which is massively expensive and gets you a quarter of the data of the twist panel. But we try to demonstrate that like what you get actually on the back end is very, very similar. And so you're estimating methylation in a very, very similar way. Even though EPIC tends to have higher coverage, but they're compressing all that coverage into a beta value. So you're losing some of that information. Okay, so if you want to employ epigenetic clocks from your methylation data, you would have your percent count or your percent methylation matrix that we got in the beginning, impute those missing values, and then you can use out-of-the-box clocks. So these are really, really easy to implement. And if you have any questions, get in touch with us. Like this is bread and butter for sure. And so Methyl Cipher is an R program or an R package that I quite like. This is coming out of the like Morgan Levine's lab and the Higgins-Chen lab. And they employ the Horvath, Hannum, like a bunch of common epigenetic clocks. Those were all based either on the 450K array or the EPIC array. And so there, like, again, we see that if you generate estimated clock values or predict age from EPIC array and the twist data, you get very, very high convergence, except for Dunedin pace. I can tell you about that later, but you get really, really high conversion. So you can use these out of the box approaches. And indeed, if you're seeing, if you have questions about epigenetic clocks or think that like the performance there isn't that good, I can almost assure you that it's not due to the twist data. It's due to either MEQTL or missingness. Like I've done a deep, deep dive on epigenetic clocks. So feel free to get in touch with me. Missingness really affects them, and MEQTL really affects between population estimates. So I won't dwell on this too long, but yeah, I'm very happy to chat ad nauseum about clocks. We are also building and like have built, it's very easy to, it's like pretty straightforward to do this, to build EM-seq specific clocks. We've done the cross-population clock across the populations I spoke about earlier. So that hopefully is coming out soon. And if you have questions about implementing clocks or building a clock for your particular population, like be in touch. It's very, very happy to do that. Okay. Something else that we that might be of interest is doing multi-omic integration. So really asking often if DNA methylation levels predict gene expression. Well, if you think about promoters, just because it's the most proximate, and typically best example. So we would expect less methylation would be associated with more gene expression. So you can just set this up in a linear modeling framework with gene expression and then percent DNA methylation. It's a very standard analysis. It's best with paired data, but you can do it without paired data as well. And then on the right is just one example of like if you chunk the promoter from distance to the transcription start site and then look at the correlation between the levels, you would expect that as you get closer to the transcription start site, you might see a stronger correlation in that negative effect. So more methylation equated with less gene expression. So it's just one like use case. The other thing you can do that's really common that people might want to do, because I think you can do it from BioView pretty quickly or pretty well, is longitudinal analyses. This is where the field is really going, especially if you're in the aging field. So this is just one example of using clocks that we made on the from the twist data, and then looking at how stable people's trajectories are across time. So are they increasing in their epigenetic age as they get older? Are they decreasing in their epigenetic age? Are they staying pretty stable? So these are just some of the things you can do with these data types that are really, I find really interesting. All right.
Yeah.
Risk scores. Yeah.
Yeah, there's like the final thing we want to talk about is just approach that we've taken in the Big Lab to make risk scores. So the caveat behind all this is that you kind of need multiple populations of data to then create the score, pick the sites, create the score, fit the score, and then apply it to see if it worked. So that might be harder if you only have BioView data, but I think there's enough data online that you should be able to hopefully do something like this if you're interested. So the way that we've done it, I mentioned we studied CHIP or clonal hematopoiesis. They are caused by loss of function or mutations, basically in enzyme that de novo methylates or de novo hydroxymethylates, which leads to demethylation often. So what we did was we just took an in vitro system. We studied what sites matter, what sites are changed when you knock out this gene. Then we calculated a score. So taking a subset of those sites, we then used elastic net regression, which is just like a way to reduce the number of features you're using. And we fit basically all the sites to predict what kind of mutation you had in the Framingham heart study. And basically we were able to shrink down from like tens of thousands of sites to just a minimum set of 800 that worked well to predict this thing that we thought was important. And then we then applied that score to enzymatic methylation data in BioView, and we were able to see how that correlated with outcomes. So then basically using, a set of CPGs that you identify, a way to fit the weights that you want to apply to those sites, and then a population to apply that score to, kind of like a polygenic risk score, or name your favorite risk score. You could then see how that performs in predicting disease outcomes in BioView. So I think that's kind of like a cool, somewhat new approach that we've been trying to work on, that hopefully will be pre-printed very shortly, for y'all to check out the actual methods and codes. But I think it's a useful framework to think about, and it's especially useful for how we can translate methylation analyses to clinical approaches.
The other thing I'll just step in and say here is that the kind of a complementary thing that our lab has also done is these are essentially epigenetic clocks, right? But you're picking your trait and training on that. So let's say you have a set of CPG sites that are age associated in one population. You could train an epigenetic clock, and then you can test in another population. Can you predict age well from that set of sites? What Yash is doing is essentially doing that, but trading out age for something else. So you can take like heart disease, and then you can ask, does this set of CPG sites predict heart disease better than this set of CPG sites? So we're happy to talk about like when and why you might want to do it, but it can be useful for comparing populations. It can be useful for comparing different sets of CPG sites. You can do this by comparing like promoters, right? They should probably predict heart disease better than quiescent region methylation. So that's kind of that. But yeah, we're happy to.
Yeah.
to talk
And then
about
I guess another, thinking really far down the line, let's say you find an amazing set of CPGs that predict breast cancer. Because it's a pro-based assay, you could then create a custom assay that only targets the sites of interest, and then that could be cheaper and easier to sequence for the future. So
That's
there are
great.
ways that's compared to if you use Epic, you'd be very fixed to that specific assay, and it would be $500 a pop. Instead, you could make your own, which is kind of cool.
Yeah, that's a really good point.
So yeah, and then we found that, I wouldn't be telling you about it if it didn't work. So it did predict low blood counts and cancer in BioView, which we thought was cool to go from like cells to EM-seq and get it to work in humans. So in theory, you should be able to do all of this now. So excited to see.
Only took us years to learn.
Yeah,
So, yeah.
exactly. So excited to see what you guys are able to discover with this approach. There is I guess we have acknowledgements. Yeah, so you guys know Angela's awesome. She will work with you on any project to get it done in the best possible way. So definitely reach out to her if you need anything. She's an amazing resource. And sort of like you want to learn from someone who helped develop the assay, which she did. So that's helpful. Same with the Leo Lab. And thanks for your time. Alex is also happy to chat about methylation. And our lab is kind of diving deep into it. And then I learned a lot of methylation stuff from the BioView team. So working on that adversity project. So wanted to shout out to them, and then you can reach out to us with any questions.
I'll also just point out that Emily Hodges, who's not on this slide, is an expert in epigenetics based at Vanderbilt. Like she's doing some really fantastic mechanistic work. So that's another lab to consider if you're thinking about really doing a deep dive in epigenetics. Emily Hodges is wonderful.
That's an oversight. Definitely should be. But yeah. And then she's done a lot of stuff requesting samples from BioView and connecting cells to like mechanisms and things like that. These are some references that we talked about. This chip methylation score hopefully will be out soon. This Longton paper is the paper that we mentioned, which compares EM-seq, or like it like introduces EM-seq as a concept, or twist EM-seq, let's call it, and compares it to Epic.
Yeah, I think Twist a few years before this put out their protocol, and then this describes like the optimization and miniaturization that was done with Vantage, and it has a lot of those Epic comparisons and RBS comparisons.
But that like describes the assay that Vantage offers, which would be the one that you may want to use, or like you will use.
Yeah.
Yep.
Cool. We do have like a CX file that we can share. Yash has put together some analysis scripts. I've put together some like very basic scripts to go from the CX file to the methylation matrices. But so we can kind of share that with you guys or do that. But also if you want to, you've gotten all the information you wanted, this is a great time to, we can kind of wrap up. Yeah,
whatever you all want. So in that directory, which is just in my GitHub, there's a CX report file that's called example CX report. It's one of the BioView ones, but I shuffled all of the counts. So it's, not real data. And then within that, there should be some scripts from, I guess I can just open it instead of describing it. So yeah. So this, great. The data is not there.
Data forthcoming.
Data is forthcoming. I can add that right now. But yeah, basically, I guess I can walk you through. So this is how we can this is a WDL, which could be directly imported into Terra.bio if you use that. And this WDL generates the matrix from the CX report file. So this is like scattering across files and basically does what we want. This is how I run logistic regression, CPG by CPG, this WDL, and this is how I run differentially methylated region logistic regression analysis. And then this is how you can run methyl CC locally with this R script, which you could take a single CX report file and estimate its self-proportions, which is probably useful to test things out. That requires this input, which is a genomic range split file. And then these two are generated from the Leia lab on how they process CX report files. So that the top two.
That's just going CX to matrix, and then I'm happy to provide any, like I work mostly with the binomial modeling, and then anyway, yeah, happy to provide any scripts. I would say between Linux and R, so or like between Bash and R.
Yeah.
So we have slightly different pipelines, but they're basically doing the same thing. So
Yep.
yeah. Cool. Okay, we can take questions or just like come around and chat, whatever.
And then there's a feedback form
Yes.
if you're willing to provide feedback. And then also will help us track attendance. So is the vibe to call it a day or for you guys to code right now? Up to you. Unclear. I will upload the data so at least you can make an informed decision. Oh, I see. It's too big to find, which makes sense. I didn't think of that, yeah. But I guess I can
Like the X chromosome or something.
yeah, I can filter. I'll just
Yeah,
stop
yeah.
sharing.
Cool.
And the recording.
Thank you guys for coming.